Hello my dear students, welcome to the MS Science Academy. Myself Meenu Srivastav, and today we are going to discuss about the cell division. The cell division, the process. What is the cell division? The process by which a cell division is a process by which cell reproduce themselves. So I will write the definition. Cell division is a process. Cell division is a process by which cell reproduce themselves. Okay, so here this in the cell division there are the two types. Okay, there are mainly two types of the cell division: mitosis and meiosis. There are another type of the cell division also, that is the amitosis, but here we will discuss only about the two types, that is mitosis and meiosis. So here the cell division, CD means cell division. The cell division is mainly two types, mitosis and meiosis. Here the mitosis is also termed as an equational division, whereas the meiosis also termed as the reductional division. I will check the camera. I have one doubt. Okay, everything is okay. So mitosis, it is known as the equational division, whereas the meiosis is known as the reductional division. Why it is named so? Because when the one cell divide into the another cells, the chromosome number will remain same. If here the chromosome number is 2n, the another daughter cells which are formed after the division, they are having the same chromosome number. As in the human cell, there are the 46 chromosomes. So after the division, after the mitosis, the same 46 and 46 chromosomes remain in the both daughter cells. Whereas in the meiosis, the parent cell will divide and finally divide into the 4 cells, and the chromosomes are reduced into half. Means if there is a 46 chromosome, then the resultant chromosomes are formed is 23. Means the final resulting cells having the half chromosome number. Okay, I have to check because I have to check the camera because I am having some doubt. Okay, so here the meiosis, so like that the chromosomes numbers are reduced. That's why it is known as the reductional division, and the chromosome numbers are equate or the same. It is known as the equational division. Here the one daughter cell, one parent cell, one cell divide into 2 cells, whereas in the meiosis one cell divide into 4 cells. Means from one cell after meiosis completely 4 daughter cells will formed. Here the mitosis is performing by the somatic cells. Somatic cells are the normal cells which are not involving in the gametes formation. Means all the cells except germ cell are known as the somatic cells. So here the cells are involved, these are the somatic cells, whereas in the meiosis the germ cells are involved which produce the gametes. Okay, but here the somatic cells are involved only for the growth and to increase the number. Okay, so here the mitosis and the meiosis. So one by one we will discuss about the mitosis and the meiosis. So in this video we will discuss about the mitosis in detail, and in the another video we will discuss about the meiosis. So here what will happen, or what are the stages in the mitosis? In the mitosis there are the two main stages. In the mitosis there are the two main stages. One is interphase. In the mitosis there are the two main stages. One is the interphase and second is the division phase. Here interphase and second is the division phase, or we can say that is the M phase. The interphase is very we can say is a time taking process, or it will take the 95% duration of the cell cycle. So it will take around 23 hours and it will take the 1 hour. Means generally a human cell take 24 hours to divide. So in that 24 hours only the interphase take the 23 hours, whereas in the division phase only the 1 hour. And what is the interphase? It is the preparatory phase. Means all the preparations will undergo during this interphase. Okay, so it is very much important to prepare the cell. All the preparations means the DNA formation, means the centriole formation, means the cell organelle formation. That preparation cell have to take and have to do, and so it will take the more time. So the interphase is the most active phase and it will take the 95% duration of cell cycle. 95% duration of cell cycle. Here the different series of metabolic changes will occur in this phase, but these changes will not visible under the microscope. So old scientists thinking that it's a resting phase, but the reality is just opposite, that it is the most active stage. So here the series of metabolic changes take place. Series of metabolic changes take place and the changes not visible under microscope. Changes not visible under microscope. Okay, so this is the interphase, and interphase is having the three main stages: G1, S and G2. Here the G1 is known as the pre-DNA synthesis phase. Pre-DNA synthesis phase. This phase take 8 to 10 hours. Means in the interphase, because in the interphase is taking 23 hours, but in the interphase the G1 is the most time taking process, because all the cell organelles will replicate in this process and all the cytosolic component will replicate. Means, so I will write all cell organelles and cytosolic component replicate. Okay, and all different types of the RNA and proteins are synthesized in this phase. Synthesize different protein and RNA synthesis. Different protein and RNA synthesis will take place in this stage. So here the G1 is a pre-DNA synthesis phase, which is the most time taking process in the interphase. And second is the S phase. S phase is known as the DNA synthesis phase. DNA synthesis phase, because the DNA synthesis will take place in this process. Okay, so replication of nuclear DNA and histone protein. Replication of nuclear DNA and histone protein synthesis in this process. Okay, so here another thing, the centriole duplication. Here I will write one, I have left one point. In the G1 phase, the centrosome replication start, but in the S phase it will complete. Okay, so here I will write centriole duplication complete. Centriole duplication complete. Okay, now second, now the G2 phase. In the G2 phase, the third one, that is a G2 phase, it is the post DNA synthesis phase. This is the post DNA synthesis phase. Here it will take only 4 to 6 hours. And S phase take 8 hours, around approx 8 hours. It will take around some 1 to 2 hours less as compared to the G1 phase. Okay, so here the G2 phase is take the 4 to 6 hours. Only some tubulin proteins and some other mitotic proteins will form in this phase. Okay, so tubulin protein and other mitotic protein synthesis complete in this process. Okay, so the interphase having the 3 phases G1, S and G2. G1 is the 8 to 10 hours and G2 is the S phase. G1 take the 8 to 10 hours. S phase, the histone protein and the nuclear DNA synthesis take place, and it will take 8 hours, whereas in the G2 phase 4 to 6 hours. It will take the 4 to 6 hours. In this phase, the tubulin and the other mitotic protein which are remaining, so they will form in the G2 phase. Okay, so here, but this is all for the interphase. In the division phase, M phase, there are the phases prophase, metaphase, anaphase, telophase, and last one, that is cytokinesis. Okay, so just take the screenshot rapidly, fastly. So here, if I will draw with a circle diagram, pie chart, if you can say pie chart. So here, what is the pie chart? If we are representing the data in a circular form. So here, like that, cell cycle. So here, G1 phase, this is S phase, 8 to 10 hours, S phase 8 hours, and G2, that is 4 to 6 hours. And here M phase, this is M phase, division phase, and here in the division phase, prophase, metaphase, anaphase, telophase, and cytokinesis. This is for, so here G1, S, G2, and this is the M phase. So this is full for the initial for mitosis and meiosis. Let's start about the M phase. In this M phase, we will discuss about the prophase, metaphase, anaphase, and telophase, and the cytokinesis. So let's get started. Now we will discuss about the M phase. M phase having the different phases like the prophase, metaphase, anaphase, telophase, and finally cytokinesis. So first we will discuss about the prophase. Okay, so what happened in the prophase? As I have described here, the early prophase and the late prophase. In the complete prophase, the chromatin fiber condense and they convert into the chromosome. Means the chromatin fibers, which are the thread-like structure, after condensing, they convert into the chromosome. So after that, the nuclear membrane disappear as well as the nucleus also disappear. So simultaneously, the centriole move towards the opposite pole and mitotic spindle start to form. So such kind of the events will take place in the prophase state. Here I will write and simultaneously you will note down. Okay, so in the prophase I am writing the combinedly, not the early phase and the late phase. I am writing the combinedly for both sub stages. Okay, so in the prophase, what will happen? The nuclear membrane and nucleolus disappear. Chromatin thread condense and convert into the chromosome. Okay, so here and the centriole move towards the opposite pole. and the another centriole moves towards the opposite pole. Centriole move towards opposite pole, and spindle fibers start to form. Okay, spindle fiber start to form. Okay, so nuclear membrane, and so like that such kind of the events will take place in the prophase. Nuclear membrane and the nucleus disappear, and centriole move towards the opposite pole, and the mitotic spindle fibers start to form. Here this is a nucleus I have formed. This is the nucleus, this is the cell membrane or the plasma membrane, and from the blue color I have drawn the nuclear membrane. Okay, so this nuclear membrane start to disappear, and simultaneously the nucleus also disappear. So this is all for the prophase stage. Now come to the metaphase. In the metaphase, all the chromosomes arrange at the mid plate with the help of the kinetochore. What is the kinetochore? I will write here. This is the centromere. This is the centromere. Here the sister chromatid, in the centromere, the sister chromatid attached with the kinetochore, like that. This is the chromosome, like that you will draw. Okay, so and this is the center part by which the sister chromatid attached to each other. This is the centromere, and inside the centromere another protein plates. This is the another protein plates also present. This is the kinetochore. This is the kinetochore. Okay, so here the spindle fiber attached here. Spindle fiber attached. Okay, here the spindle fiber attached. This with the help this kinetochore. With the help of this kinetochore, and this spindle fibers just arranging the chromosome on the metaphasic plate. Okay, so here I have drawn here some chromosomes attached to the spindle fiber with the help of the kinetochore and arranged at the mid plate. Okay, so I will write. I will write the complete discussion about the metaphase. First you will write it, or either take the screenshot so that I can get the space, because the space is not that much sufficient. So that's why I have to erase it out. So here I am erasing. This is all for the prophase, and now come to the metaphase. The metaphase, the chromosome arranged at the equator. Chromosome arranged at the what I am writing. This is the metaphase. Chromosome arranged at equator. Chromosome arranged at the equator on the with the help of the spindle fiber. Okay, chromosome arranged at the equator. This is called metaphasic plate. This is called the metaphasic plate or mid plate. Okay, so here the chromosomes are easily identified in this stage. So if anyone wants to study the chromosomes with the help of the microscope, this is the best stage to study the chromosome. Some if in the MCQ if they will ask, then which stage is very much prominent in the for the study of the chromosome, so you will take the metaphase, because the chromosome here arranged in a very proper manner and very condensedly they are arranged, so it is very easy to study the chromosome. So in this metaphase, this is the best stage to study the chromosomes. Okay, now come to the anaphase. What is the anaphase? Anaphase is the shortest stage. Here the centromere is split on the kinetochore part. I have discussed what is the kinetochore. In the centromere there are the two plates. These are the kinetochore. Okay, and the sister chromatid attached with the help of kinetochore. So the spindle fibers continuously stretching the sister chromatin with from this kinetochore, and the kinetochore split it out and the sister chromatid separate from each other. Okay, so here the centromere is split and the sister chromatid separate from each other. That's all for the anaphase. So this is the shortest stage. So I will write here the anaphase. I will write here anaphase. What will happen? This is the shortest stage. First thing, shortest stage. Chromosome split. Chromosome split into sister chromatid and sister chromatid, chromosome is split into the sister chromatid from each other. That's all. So the anaphase is the shortest stage. Now come to the telophase, as here the chromosome movement from because the stretching power of the spindle fiber, these spindle fiber taking the chromosome at the opposite poles, okay, one on this pole and the second on the another pole, and the reverse prophase stage will start. Means the telophase is nothing, it is just the reverse prophase. It is just the reverse prophase. What will happen in this? I will erasing, just take the screenshot in the metaphase and the anaphase. So I will erase it out so that I will get the space. Here what will happen? The telophase, what will happen in the telophase? The sister chromatid are attached, the sister chromatid reaching towards the opposite pole and uncoiling, and we can say the uncoiling of the chromosome means the chromosome convert into the chromatin fiber, and the nuclear membrane and the nucleus reappear. Means all the events which have done in the prophase in the telophase, all the events of the prophase just opposite. Okay, in the prophase the nuclear membrane and the nucleus disappear, but whereas in the telophase the nuclear membrane and the nucleus reappear. In the prophase the chromatin fiber convert into the chromosome, whereas in the telophase the chromatin fiber reappear. Means the chromatin thread, chromosome will convert into the chromatin fiber. Okay, so like that. So this is the telophase is the reverse prophase, prophase. Here chromosome movement stop, chromosome movement stop, nuclear membrane and nucleus reappear, chromosome convert into chromatin fiber, again thread like structure. So like that the telophase completed. Here the cytokinesis will start. Cytokinesis means the cell division. What will happen in the cytokinesis? In the animal cell, the cytokinesis proceed with the help of constriction and the furrow formation. Actin microtubules, just they are arranging inside the plasma membrane and they are forming a contractile ring, and that pull the plasma membrane progressively inward. Okay, so here what will happen in the cytokinesis? Cytokinesis, okay, so we will write the cytokinesis is done in the animal cell especially by the constriction and the furrow formation. Whereas in the plant cell cytokinesis differ. Okay, in the plant cell it is formed by the cell plate formation, but whereas in the animal cell the cytokinesis is formed by the constriction and the furrow formation. Okay. Okay, so cytokinesis by constriction and furrow formation. Constriction and furrow formation. Actin microtubules form a contractile ring which drag the plasma membrane inside towards center. Okay, so here as I have shown in the telophase, because the cytokinesis will start from the anaphase itself, not in the late anaphase. So, but I have drawn here in the telophase that the cell membrane will just reverting toward forwarding towards the center, and after completing the telophase, this cell membrane will continuously moving towards the center with the help of the actin microtubules or actin microfilaments, and by this complete ring form the two cells. Okay, here this is the nucleus. Okay, and in the cytokinesis the complete two cells are formed from the one cell. Here I have taken the one cell, and after the complete cytokinesis the two cells is formed. This is whole for the mitosis. Okay, after in the next video we will discuss about the meiosis.